In an earlier presentation, we saw that after glycolysis and 2 turns of the Krebs cycle in aerobic organisms, the free energy that was once in glucose is in 10 NADHs, 2 FADH2s, and 4 ATPs. So in this module, we'll see how the free energy in the reduced electron carriers is released during electron transport and captured in a gradient of protons. In a later module, we'll see how relief of this proton gradient is coupled to ATP synthesis by oxidative phosphorylation. First, a reminder about redox reactions. In this generic reaction, electrons are being transferred as part of hydrogen molecules, a characteristic of many biochemical redox reactions. The reduced and oxidized forms of the same molecule are called a redox pair. From the equilibrium arrows, you can see that this reaction is exergonic, so it would have a negative standard free energy change, a negative delta G0, releasing free energy. How about this real reaction? Can you identify the redox pairs here? They are pyruvate and lactate, and NAD+ and NADH. Pyruvate and lactate are simply the oxidized and reduced forms of the same molecule, as are NAD+ and NADH. The transfer of electrons between molecules in redox reactions is a lot like the flow of electrons in an electric circuit. Take this simple battery, for instance. By convention, the plus button, or the plus end, is the anode, and the flat end of the battery is the cathode. When the circuit shown here is closed, electrons flow from the cathode to the anode. This is because the anode contains chemicals with a higher potential to be reduced than the chemicals at the cathodal end of the battery. The chemical reaction in the battery is spontaneous, releasing free energy. The electron transport system, then, is a series of molecules in the crystal membrane of the mitochondrion that can accept and then donate electrons. The electrons flow from NADH and FADH2 down this series of redox molecules because each subsequent component has a higher potential to be reduced than the one before it. Hence, the whole of electron transport releases free energy when the reduced electron carriers produced in glycolysis and the Krebs cycle are oxidized.